Okay, so for Form 3 students, English class, for homework on the 15th of May, I would like to make a solution video here for your reference so that you can know and know how to answer the questions. Well, the first question, there are 10 multiple choice questions here. So I want you to understand how to answer these questions. And also one long questions. What is anabolism? Anabolism is part of all metabolism. So anabolism means the building up actions. Please see the right hand side. From the simple molecules to a complex bigger molecules which require energy. This is a building up action, okay? Building up action. Let me draw something. Is all the building up actions in an organism. Okay. So the answer is C. The answer is C. Question 2. Oops. Question 2. About enzyme, which of it is correct? So let's take a look at the several properties of enzyme. Properties of enzyme. Okay, the actions and properties of enzymes. So enzymes will fit with the substrates, combine with it, reduce the activation energy. So there are, and we use lock and key hypothesis to explain that. So let's take a look at the questions. The enzyme is not correct. See which one is not correct. It can be affected by temperature and pH. Yes, temperature and pH. When temperature and pH is, enzyme is a protein, is a type of protein. It can be denatured under high temperature. Can be denatured under high temperature. Or the highly acidic or alkaline conditions, that is what we mean by pH, it will denature. So to affect the activities. This is correct. It only works in the bodies of organisms. No, for so many experiments in the videos I show you, it is performed outside the organism. It can be performed in a plastic bag, can be performed in a test tube, etc. So only works in this organism. Let me erase that. It is not correct. Not only work in that. B is the answer. The specificity of the actions can be explained by lock and key hypothesis. So take a look here. By lock and key hypothesis, we mean that only one enzyme, one key can open that lock. Other key cannot open that lock. And they help to speed up reactions in organisms. Yes, that's correct. So 3 of them are correct. But I also want you to know why the change in temperature and pH would affect their activities. When when temperature is too high, or when temperature is too high, or when pH is not correct, you see that the shape, the active site of enzyme is changed in shape. So just like a key， 當你磨平晒啲鎖匙嘅痕之後，嗰啲齒啦鎖匙 ，when you remove all the shape from a key, that key can no longer open a lock. So it's same as the denatured enzyme. So the graph shows the amount of products in an enzymatic reactions. So the effect of adding an inhibitor. Initially, you should see the reaction rate is quite high. Along the time, there's more and more products accumulated. Accumulation, that means 積聚. With inhibitor, then of course the reaction rate is slower. So you see the slope is less steep. For example, like this, less steep. But what is the answer? At last, you should see that both setup with or without experiments, their final amount of products should be the same. Since we have the same amount of substrates, if we have same amount of substrates, then the enzyme just convert them slowly when inhibitor is added. But at last, everything is converted into products, so that the amount of products should be the same. So the answer is B. Question 3 is B. Question 4. Question 4. We have a product. Oops. We have a starch agar blocks. Starch agar blocks. Doesn't matter with the agar blocks as long as it is something about starch. Okay. So one tube contains fresh enzyme. When the fresh enzyme is present, they convert. The fresh enzyme is capable to convert starch into sugar. When starch is present, iodine solution iodine solution can turn starch into blue-black color. Okay, that's blue-black color. So you see that it means that when fresh enzyme solution is present, blue-black color is not present. It is completely converted into sugar. Starch is no longer present. Starch is when enzyme is present, it converts all the starch. So there's no blue-black color. On the other hand, when we have the boiled enzyme solution, then boiled enzyme solution, all the enzyme does not work. They do not convert the starch into sugar. So all the starch will give you blue-black color. This is what the experiment means. So possible explanation of this results. Of course, the enzyme, when enzyme is present, you see that? When the enzyme is present, they can help to break down the starch. When enzyme is boiled, they are denatured. They cannot break down the starch, so that you can see blue-black color is here. The answer is A. The enzyme may be an amylase. In this case, amylase is something specific, something special. amylase enzyme breaking down starch. Okay. I want you to remember that whenever you see the suffix, the word, the letters ASE, that's something enzyme. For example, lipase is enzyme breaking down lipids. Or another case, protease enzyme breaking down protein. Okay, so remember the naming method. And question, so the answer is B, 5B. Question 6, in very hot water, high temperature, so the enzyme is denatured. You see that when temperature increases further beyond the optimum temperature, enzyme is denatured. So no question about this, 6D. Okay, so question 7 shows the possible substrate of this enzyme. As long as the substrate can, as long as the active site can fit with the substrate, then this is a good substrate. This can be a good substrate, this can be a good substrate. All of them can be a good substrate. So the answer is D. 7D. They can fit into the active site. Just like a key can be fit into a lock. So it is a good key. A good lock to be exact. Shows a box of contact lens. Pay attention. They want to remove the protein deposits. Remove some proteins. So the active ingredient, the enzyme, is I use cross. Active ingredient, it is not a lipid. Enzyme is a protein, okay? Enzyme is a protein, as I mentioned. Enzyme is a protein. On the right hand side, you can see it is reusable. Good. It is reusable. At the right hand side, can be reused. The action is specific. Specific action. Take a look at this. This is one of the important characteristics of enzyme, specific action. So the answer is C. Question 8. Question 8 is C. Question 9. So to investigate the effectiveness of biological washing powder in removing different stains. Removing different stains. They want to check different stains. So we should take a look to look for different stain. For example, lipid stain and protein stain. Okay, this should be the only thing to change. Lipid stain versus protein stain. And then the other factors has to be kept constant. You should not change other things. For example, these both are using biological washing powder. For example, however, the setup 1 is using non-biological washing powder, so it's not correct. And also, setup 4 is using 35 degrees, different temperature. This is also wrong. So the answer for question 9 should be comparing 2 and C. The answer is D. Okay. Last MC. Which of the following factors is under investigation when I'm comparing cell 3 and 4? So take a look at 3 and 4. What is under comparison? What is the same? Protein is the same. Okay. What else is the same? Biological washing powder is the same. Biological washing powder is the same. And the difference, and the only difference, lies in 35 degrees and 25 degrees. So the temperature is different. So we are investigating the effect of temperature. Okay, you see that the answer is A, temperature. We're checking the temperature. Okay. Question 11. Complete the table. Amylase, as I mentioned above, amylase is actually catalyzing the breakdown of starch into maltose. Maltose is a sugar, which is sweet. This is amylase, 澱粉酶 in Chinese. Okay, the second point is the ingredient of washing powder. Let's take a look at the PowerPoint here. Biological washing powder, they contain, what do they contain here? They contain protease to break down protein. They contain lipase to break down lipid. Okay, protease and lipase. The protease and lipase helps to break down insoluble proteins and lipids into soluble products. When they are converted into soluble products, so that it can be washed away by water. Okay, last one. Meat tenderizer, that is 鬆肉粉. Meat tenderizer contains an enzyme called papain or protease. You can also call it protease. Protease helps to break the proteins in meat, so that the proteins becomes less tough. So the meat becomes soft. Okay, good. Last question, part B. Why would the effectiveness of an enzyme containing washing powder reduce in hot water? So give an example with reference to the active site. I want you to take a look. I want you to take a look at the right-hand side. The enzyme in washing powder would be denatured under high temperature. Would be denatured under high temperature. Okay. Enzyme is denatured. What does it mean by denaturation? It means that the active site of enzyme is changed in shape. You can see that green part. The active site is changed in shape. So the substrate molecules in purple, it can no longer fit into the active site, so the enzyme does not work. Okay, denature, first point. Second point, shape of active site is changed, and the substrate molecule can no longer fit into that. So the enzyme does not work. This is a perfect answer. Okay, so this is all the solution I have for this homework on the 15th of May. Please remember to submit the homework and ask me questions.